Excellent. So thank you, Esther, for the kind introduction. As you mentioned, I'm a bioinformatician, and I'm more than happy to provide any advice or support in these cohorts, because bioinformatics is getting traction and a lot of projects are using bioinformatics, and it's not an easy skill. So just let's get into it with a bit of background. So breast cancer research is needed. And why is that? Because it's the most common cancer in women worldwide, because approximately like 670 women died of breast cancer in 2022, and every year is a similar outcome. In Australia, 9 people die from breast cancer every day. And recent statistics show that diagnoses have increased by 24% over the past decade. So this really highlights that cancer research is needed in the breast area to improve patient outcomes. And for me, a great opportunity to thank Cure Cancer and NCC Johnson for the support to achieving this goal. So what is the actual problem? The problem is that breast cancer is an heterogeneous disease. We have multiple subtypes of breast cancer, of which I'm focusing on the HER2-positive. The HER2-positive disease encompasses roughly 15% of the total breast cancer tumors and is characterized by the production of the HER2 protein. And that production of the HER2 protein leads to increased proliferation and cell division. So it's an aggressive phenotype. So just to show a cartoon depiction on what is the nature of HER2, in the normal cells, we have the HER2 receptor in the cell surface of the cells. And the role of this molecule is to activate signaling pathways that command the cell to survive and proliferate. So cancer cells exploit this property by over-amplifying and enriching the receptors in the cell surface by gene amplification of the HER2 gene. And therefore, they really have achieved this aggressive phenotype. And we see that clearly from immunohistochemistry staining. And here you can see high amplification of the HER2 gene under the microscope. And we can also see it genomically. So when we evaluate the status of HER2, when it's amplified in this cohort, the MetaBrick, evaluating around 2,000 women with breast cancer, we corroborate that HER2, when it's amplified, is also overexpressed. So this really shows that the biology of this tumor really depends on HER2, and we call that it is an oncogene-addicted tumor. So what is the treatment of HER2 therapy? So initially and originally, this tumor had a poor prognosis. And the reason of that was that there was no target to the HER2 gene. But it was until 1998, with the introduction and discovery of trastuzumab, which is a molecular antibody that binds to the HER2 receptor, that the downstream signaling is suppressed and the tumors are stopped, and therefore the survival of these patients is starting to increase. Since then, multiple combinations to anti-HER2 therapy have been evaluated, and now the landscape of HER2 positive breast cancer is very promising. However, there are still challenges, otherwise I wouldn't be here giving this talk. And what are those? So long-term use of anti-HER2 therapy will lead to resistance. So just to give you an idea of the numbers, around 20% of the patients that are diagnosed early stage will progress to advanced lethal disease with some degree of resistance to the HER2 therapy. Of them, 50% will develop brain metastases that are associated with high mortality, with less than 2 years survival, and really a poor quality of life due to the damage of the central nervous system. So this is just, and also it's very important to highlight here that when we are profiling tumors that are addicted to a very particular oncogene, by characterizing the resistance to this, really opens computational approaches that we can apply to different tumors that are more heterogeneous. So what is the landscape, genomically speaking, of primary tumors that are HER2 positive? And for that, I'll bring the Affinity trial that I did some analysis on this. The Affinity trial is a phase 3 adjuvant study investigating the benefit of pertuzumab with chemotherapy that enrolled 5,000 patients to this study. In here, we selected 950 patients for targeted DNA and RNA sequencing. So what do we find by characterizing primary HER2-positive disease. In the landscape of point mutations, what we identify is like alterations of the PI3K AKT pathway, which is a pathway that interacts with HER2. When it is altered, it really associates with adverse outcomes. And we hypothesize that it's becoming independent of the HER2 signaling, so the suppression of HER2 is not effective anymore. In the copy number landscape, I just want to highlight there was no big surprise. We found MYC alterations. MYC is a very poor prognostic marker when it's amplified not only in HER2, but in other malignancies. And interestingly, TOP2A, which is a gene that is very close in proximity with the HER2, when it's amplified, it seems to be associated with favorable outcomes. And we think that is the way that HER2 is breaking when it's trying to amplify that leads a specific pattern. So these breaking points on HER2 are interesting to characterize because not all of them might be functional, and therefore there might be some dynamic happening there. So what do we find? What do we find in characterizing the transcriptomic landscape of primary malignancies? So we use this 3-gene signature to measure activation of the immune system by measuring effector signaling of T cells. And what we found when this is the combination of these three genes are overexpressed, we really have favorable outcomes for these patients. So key message here is the primary disease of HER2 in the molecular landscape is suggesting us to really look at the mutations and also look at the immune system and the interplay that are between them. So this brings me to the Cascade study, which is Peter Mack's post-mortem autopsy study. And let me give you a brief overview of this project. So patients are originally diagnosed with breast cancer, and then we extract the primary tumor during surgery, and these tumors undergo DNA and RNA sequencing in a whole genome assay around 120x, which provides enough depth to really characterize the heterogeneity in these tumors. So these patients are enrolled for standard of care, or some of them really are enrolled in clinical trials. And if the patient relapses, we monitor and take continuous blood samples to measure residual disease. And when the patient dies of metastases, we have a rapid autopsy and collect these biospecimens that are cryopreserved and then sequenced, again, by a whole genome and RNA assay. So usually we take around 10 samples for sequencing, and we collect another 20 for downstream analysis for validation or by PDX. So this pilot study has two arms. So we are first evaluating 6 patients that have received standard anti-HER2 therapy, trastuzumab, pertuzumab, T-DM1, a combination of them depending on how the patient relapsed and the recurrence emerged. Interestingly, 3 of them receive immune checkpoint blockade, so we can really see how immunotherapy changed the mutational landscape of these tumors. And as a second arm, we have patients that receive new agents like T-DXd and tucatinib. So overall, this study is powered to really reveal the resistance mechanisms of new therapies, from trastuzumab to really new agents as tucatinib and T-DXd. So where are the samples that we collected? Most of them collect from the brain, lung, and liver. As Kelly mentioned in her talk, those are common sites of metastatic deposits. We have matched primary, and if in the autopsy report during the surgery, it's highlighted that a certain area has contributed to the death of the patient, we sequence that as well. And again, this plot comes from a review from different autopsy programs across the world. And for me, just to highlight that in Cascade, we achieve rapid autopsies, that we are collecting all the tissues within 6 to 7 hours from patient death. And that allows to have a unique quality that is the best for research. And also this also is a great opportunity to acknowledge the patient, that their contribution to create this compendium of advanced malignant tumors that we can study in a greater detail. And therefore, although cancer took the lives of these patients, their fight against cancer carries on with this initiative. Which brings me now to the study aims. We want to characterize the mutational transcriptomal landscape in the metastatic setting and contrast that with the primary setting like we did in Affinity. We have specific questions to know if HER2 mutations or key signaling pathways are driving therapeutic resistance, or and we want to know what additional driving mutations emerge after primary tumor and that are associated with therapeutic intervention. Naturally, we want to characterize the immune system as well, just to have a rationale for immunotherapy in these cases by evaluating the prevalence of neoantigens, and if there is genomic events, diluting that exposure of neoantigen. And of course, we want to establish the integrity of the HLA, the MHC class 1 antigen presentation pathway, establishing HLA loss, repression, or aberrant splicing, which kind of disrupt the capability of the immune system to recognize these cells. And finally, we want to establish what are the genomic profiles of those clones. We want to know what is the genomic composition and know what is, if there is a common mutations, and actually when they emerge in cancer evolution. So I can share some preliminary results from this study, and I can say that the metastatic landscape is highly heterogeneous. Let me give you an example of that. And here we have a primary sample. This is a circus plot if you're not familiar with that. This is the whole genome from chromosome 1 to chromosome X. And in here in the second circle, we have the point mutations of this tumor, this primary. And here in the green, we have the copy number alterations. As you can see, HER2 is very amplified, and the area around HER2 is amplified. And in the inner circle, we have the rearrangements. So when we move and we observe the metastatic samples, we see an extensive shift from DNA alterations. We see a massive increase in point mutations, copy number, and structural rearrangements. So this highlights the work for many others, from Peter Nowell to Charles D. Swanton, and many others in between, that metastatic is a multi-step process fueled by genomic instability. And this is a common denominator that we see in all the cases of Cascade, and it's also been published by colleagues from our lab, from our previous autopsy report on breast and luminal and triple negative tumors, and from Cascade from melanoma and ovarian malignancies. And just to start wrapping up, I want to really show you an evolutionary reconstruction of a patient. So this patient was diagnosed of cancer at 36 years old and had an overall survival of 6 years. This patient had immunotherapy as this person enrolled into a clinical trial that was evaluating immune checkpoint inhibition, and along the way has multiple anti-cancer agents. So when we sequence the primary tumor, we observe, of course, amplification of HER2 with key drivers like p53. So this is a bad starting point. And in here, you will see, I'm showing here the expansion of this subpopulation. So we have the primary diagnosis. And when we start evaluating the samples, we see a massive, interesting shift. So the second event that we see is LOH on p53 and a reduction of the amplification of the HER2. So HER2 has a reduction, let's say it's approximately from 80 to 90%, is becoming de-amplified. And this subtype is transitioning to a different subtype, more like a triple negative. This was accompanied by an enrichment of Applebeck signatures, which highlight the mutagenesis that is happening here. And the invasion of proximity lymph nodes. So the first lymph node to be invaded was the para-aortic, which is in the chest wall. And we see that in there, the tumor acquired really deleterious focal copy number amplifications, which involve MYC and KRAS, which is very unique for a cohort. And we kind of correlate that the emergence of this clone was at this point. There is no evidence for this patient, but this patient has already, the clonal expansion is already happening, but there is no evidence of this disease on this patient. So after that, we see further progression of this tumor, and the invasion went to the lung. In this area, the lung recurrence, the patient was diagnosed with the recurrence at this site, and the treatment changed completely to have different types of immunotherapy, and then it was rectified that this tumor was behaving more like a triple negative. So anti-HER2 therapy was not effective anymore in this case, and therefore it continues its progression to the liver and then into the pericardial area, and altogether contributing to the death of this patient. So this is just to highlight how powerful is integrating all these samples to really trace the evolution of this tumor and see these dynamic changes as they unfold on the timeline. And we are able to track when is this lethal clone emerging, and we are able to measure its composition, which is loss of HER2, TP53LOH, MYC, and KRAS amplified, just to give you some key drivers in this component of this tumor. So key takeaways from this study. Autopsies really overcome some sampling limitations that we see on the primary samples, that we don't have enough biomaterial to sequence. And it also reveals the landscape of tumors that is very metastatic, it's complex. But by integrating all these samples, we are able to characterize what's going on. And importantly, by really integrating these autopsy reports with the timeline and having other measurements of disease progression, we are able to disseminate the routes of invasion of these tumors and really corroborate the key driver mutations in this multi-step process of metastatic dissemination, and what is this tumor doing to break these barriers to, adapt to foreign tissues, really overcome these anti-cancer agents. And therefore, we have clinical implications. One of the key objectives of Cascade in the long term is to inform if immunotherapy or HER2 targeted therapies, how to really do therapeutic shifts and try to monitor on residual disease in blood to get ahead of the tumor before it's too late. And with that, I'd like to thank the Loy Laboratory, especially the people involved in my lab on the on Cascade, which is Peter, Joyce, Roberto, Corny, the Cascade team, the people taking the biopsies, patient advocates, the bioinformatics core, and something that I didn't have time to touch on, but this project is in collaboration with the Crick Institute, with Charlie Sawanton Lab. I have a lot of input from Charlie, from Nicholas, from Chris Bailey to measure some of these HER2 changes that are probably breaking points by extrachromosomal DNA, and therefore we are able to see this dilution of that on oncogene. And with that, I'm happy to take any questions.